Welcome back to Chapter 3, Amino Acids, Peptides and Proteins. In this lecture, we'll be looking at Part 2 of this chapter. Here, we'll learn about unnatural amino acids and we will extend our conversation on amino acid ionization and look briefly at peptides and proteins. This slide revisits all the 20 amino acid structures and their names. Now, I cannot emphasize more on how important it is for you to remember and relate the name and structure when it comes to amino acids, because amino acids are the basic building blocks and basic building blocks of proteins, and we will revisit the name of these amino acids again and again throughout. So this is the time for you to remember. What you need to remember absolutely is the name of the amino acid and the one-letter abbreviation. And if you can, relate the name to the structure. If you cannot, at least remember the groups, right? There's non-polar aliphatic R groups, aromatic R groups, the positively charged, negatively charged, and the polar uncharged ones. The positively charged ones and the negatively charged ones should be straightforward. You should be able to remember these two and the aromatic ones. Some of the polar uncharged and the non-polar aliphatic groups are a little difficult to remember, but once you practice, you should be able to remember them as you go along, right? So remember, 20 amino acids and pKa values. 7 amino acids have pKa's. And pKa's for side chains, right? Now, talk about side chains: tyrosine, lysine, arginine, histidine, aspartate, glutamate, and cysteine. 1, 2, 3, 4, 5, 6, 7. There are 7 amino acids with side chain substituents. These are the side chain substituents or the R groups that has a pKa. In addition to these 20 naturally occurring amino acids, there are non-natural or non-canonical amino acids as well. There are more than 300 other amino acids found in cells, greater than 95% of which don't get incorporated into proteins. Some notable and common amino acids found in proteins are 4-hydroxyproline and gamma-carboxyglutamate. So how do these non-canonical or uncommon amino acids get incorporated in proteins? These amino acids are not incorporated by ribosomes, except for 2, which are pyrrolysine and selenolcysteine. Now remember, amino acids are the basic building blocks of proteins. Proteins are polymers of amino acids that are attached covalently to each other, right? And the covalent attachment is performed by ribosome, right? Remember your transcription and translation machinery. Now, the way these uncommon amino acids get attached to proteins is by a process named post-translational modification. Proteins, after they get translated by ribosomes, get acted upon by specific enzymes that modify these amino acids, and hence the name post-translational modification of proteins. These modifications can be reversible. An example is phosphorylation, and this modification is important in regulation and cell signaling. We'll talk about this a little more as we go along. Selenolcysteine is an analog of cysteine. The only difference is that the SH group is replaced by SEH, and it is not coded for directly in genetic code. Remember, the genetic code or codon makes proteins, right? Each amino acid is coded by 3 nucleotides, which are called codons. Special encoding is used, and that is UGA. Normally, it's a stop codon, but it is used by ribosomes to code for selenolcysteine. It is known as a part of about 25 human proteins. Selenol group has a pKa, a lower pKa than cysteine or a thiol group, and so selenol would have the side chain of selenolcysteine would have a pKa lower than that of the side chain of cysteine. And cysteine, remember, has a pKa of close to 8. Deprotonated at physiological pH. So this H would be lost at around pH 7 for selenolcysteine. Selenoenzymes use selenolcysteine in catalytic processes. So that's one of the key reasons why selenolcysteine is incorporated in proteins. Another classic example of an amino acid modification or a post-translational modification of a protein is methylation. Methylation is the process of adding a methyl group onto specific amino acids in proteins. For example, arginines in proteins can get added with 1 or 2 methyl groups. Lysines can get added with 1 or 3 methyl groups, right? And what are the most common elements of these amino acids? And if you look at the structure, one of the most common element is the amine group, right? The NH3 plus here and the NH2 plus or the NH3 plus in arginine. The enzymes are methyltransferases. They transfer a methyl group from a cofactor to the substrate protein, whatever it is. Histone methylation is a classic example and is important in epigenetics. It's associated with gene expression, right? In this case, downregulation of gene expression when it's methylated. So we'll talk about this in detail in advanced biochemistry if you are indeed taking this course. But for the purpose of this course, you only have to remember that methylation is an amino acid modification. Acetylation is another amino acid modification, right? Majority of eukaryotic proteins have N-terminal acetylation. Enzymes that are involved are N-terminal acetyltransferases. They are critical for function and regulation of many proteins. Their stability and localization. Histone acetylation is important in epigenetics and is associated with an upregulated gene expression. The enzymes that are involved in histone acetylations are histone acetyltransferases. And the enzymes that remove these acetyl groups are histone deacetylases. So if you were to look at this figure that is shown here, a lysine side chain gets acetylated by histone acetyltransferases, where it adds an acetyl group to the side chain amine. Whereas histone deacetylases remove this acetyl group and brings back the original protein amino acid, that is just a lysine. Now, these acetyltransferases and deacetylases are called as readers and erasers, right? So because they are associated with gene expression, these enzymes regulate gene expression by regulating the acetylation status of histones. Now, you have to remember that acetylation is a post-translational modification. One of the key things that acetylation does is it removes the charge from the from the side chain of lysine. If you look at here, lysine is an amine. Once it gets acetylated, it's an amide. Amide doesn't have a charge. So that's one of the most important things that acetylation does. Methylation will not remove the charge, right? If you add methyl groups, the charges may still persist. The third key amino acid modification is phosphorylation. Phosphorylation is a process wherein an enzyme adds a phosphate group to either a serine, threonine, or a tyrosine amino acid residue on the protein, right? And these enzymes are called as kinases, and protein kinase to be specific when the substrate is a protein. They use ATP as a cofactor and transfers a phosphate group from ATP to either a serine, threonine, or tyrosine hydroxyl group. If you go back and look at the structures of serine, threonine, tyrosine, the side chains have a hydroxyl group. That's the hydroxyl group that gets modified to a phosphate. Kinase phosphorylate and phosphatases remove that phosphate, and they do this by a hydrolysis reaction. H2O is being used to remove the phosphate and bring back the hydroxyl group of the original protein. Like I said, ATP or GTP donate phosphate group for kinases. And kinases typically require divalent cation like magnesium to stabilize a reactive phosphate group. Now, the whole process is for regulation of various protein function. For example, a protein substrate, when get phosphorylated, can either get turned on or turned off. And we're talking about the activity of that specific protein, right? And specifically in terms of regulation of cellular function. Often, phosphorylation serves as a switch between active and inactive forms. In addition to these 3 amino acid modifications that I just talked about, there are many other amino acid modifications that takes place in the cell. Some other examples are hydroxylation, where lysine and proline residues, particularly relevant in collagen, gets hydroxylated. Enzymatic action requires vitamin C. So an enzyme that does hydroxylation uses vitamin C. So if you have vitamin C defects, you get scurvy because connective tissues are affected. The reason is because lysine and proline in collagen cannot get hydroxylated, and as a result get tissue defects. That results in scurvy. That's why you need vitamin C. Glycosylation is another example. Glycosylation is a process by which proteins, sugars are added to proteins, carbohydrates. Now, there are N-linked and O-linked glycosylations. Variation, glycosidic bond linkage, glycan composition, and structure are present. It affects protein folding, stability, and cell-cell adhesion. If a protein that requires to be glycosylated does not get glycosylated. We'll learn more about glycosidic bond linkage, glycan composition, and glycosylation later on when we talk about carbohydrates. Lipidation. Myristylation, palmitylation, and farnesylation are some of the examples of lipidation. And in this case, proteins get modified by lipids. And why is this important? Because protein localization and function depends on these. Finally, deamination or protein citrullination and deamidation. Deamination of arginine residues by this enzyme, protein arginine deaminases, results in the conversion of arginine to citrulline. This is an important modification because it removes the charge from arginine and makes the protein surface neutral. Deamidation of asparagine and glutamine to aspartate and glutamate adds charges. Remember, asparagine and glutamine are polar uncharged amino acids. Enzymes that hydrolyze these amides to carboxylates will add charges. In this case, adding a negative charge, right? So these are some of the other amino acid modifications. Now what you need to remember is what these different amino acid modifications are. You do not have to read in detail. Let's take a step back and look at the acid-base properties of amino acids again. You know that amino acids have ionizable groups, and as a result, they will have specific titration profiles or titration curves. These titration curves will tell us as to what charge state an amino acid will be at a given pH. The pH at which the net charge is 0 for an amino acid is known as the isoelectric point of that amino acid. You can also extend this definition for a peptide. Isoelectric points for amino acids will differ depending on their side chain structure, if they are incorporated in a protein, and their local environment. Let us consider the acid-base titration curve for glycine, which is a simple amino acid that is devoid of any side chain substituent. The two ionizable groups of glycine, the carboxyl group and the amino group, are titrated with a strong base such as sodium hydroxide. And so, as you remember, the titration curve has pH on the y-axis and the OH minus equivalent on the x-axis, OH minus coming from sodium hydroxide. The plot has two distinct stages corresponding to deprotonation of two groups. One is a carboxylic group and the other one is the amino group. Right? Now, each of the two stages resemble in shape the titration curve of a monoprotic acid, such as acetic acid. And if you remember the acetic acid titration curve from Chapter 2. Now, let's begin. At low pH, the predominant ionic species of glycine is this one, and it is positively charged because there's a positive charge on amine and no charge on carboxylic acid. So it is fully protonated. In the first stage of the titration, the carboxylic group of glycine loses its proton. So this is the proton donor in this case. And as we increase the OH concentration, OH minus concentration, the carboxylic acid loses its proton, and at the midpoint of this stage, equimolar concentrations of the proton donor and the proton donor, which is this, and the proton acceptor, which is this group, are present, equimolar, at the midpoint. Now, if you remember, as in the titration of any weak acid, a point of inflection, and that is the point of inflection, which is the midpoint, is reached where the pH is equal to the pKa of the protonated group being titrated. So this is the first inflection point, and this is where the pKa is equal to the pH. For glycine, the pH at the midpoint is 2.34. Now this is labeled as pK1, or it is the pKa of this carboxylic acid group of glycine. Now if you recall what you learned in Chapter 2, pH is a convenient notation for proton concentration. Remember, right? pH is minus log H plus, right? pKa, on the other hand, is the equilibrium constant for ionization. Now, what is pKa? If you remember, pKa is a measure of the tendency of a group, in this case, tendency of a carboxylic acid functional group, to give up a proton, with that tendency decreasing tenfold as the pKa increases by 1 unit. So if a pKa goes from 2 to 3, it means that it has increased tenfold because it's in logarithmic unit. Now, after this, as the titration of the glycine proceeds, you're adding more and more sodium hydroxide, and as it proceeds, another important point is reached, and that important point is at pH 5.97. If you look at the pH, it is 5.97. Now, here there is another point of inflection, if its removal of the first proton is essentially complete, which means that this proton is now completely removed to form COO minus and NH3 plus. At this pH is 5.97, glycine is present largely as the dipolar ion or Zwitter ion, as we call it. Now, this point is called as the PI. Right? Now, we talked about PI, and we'll talk about this again in the next slide. The second stage of the titration, if we continue and get to the second stage, corresponds to the removal of a proton from this amino group. NH3 plus gives up a proton. The pH at the midpoint of the stage is 9.60, which is equal to the pKa of NH3 plus, or labeled as pK2. The titration is essentially complete at pH of about 12, at which point the predominant form of glycine is this, which has a negative charge. Now, from the titration curve of glycine, several important pieces of information can be obtained. Right? First, it gives a quantitative measure of the pKa of each of the two ionizing groups. First, pKa of carboxylic acid, which is 2.34, and pKa of the amino group, which is 9.60. Right? Second, it also tells us that this is a buffer zone for the amino acid. So essentially, there are two buffer zones. Then one is between, so it's at pH 2.34 plus or minus 1, right? So glycine is essentially a buffer at this buffer zone. Amino acids carry a net charge of 0 at a specific pH, and which happens to be the PI. In the previous slide, PI of 5.97 is when the net charge of glycine is 0, because it has a positively charged amine and a negatively charged carboxylate. So the charge cancels out. Zwitter ions predominate at pH values between the pKa values of amino and carboxyl groups. For amino acids without ionizable side chains, the isoelectric point is Pi is equal to pK1 plus pK2 divided by 2. At this point, the net charge is 0. Amino acid is least soluble in water. Why do you think so? Just give a thought. Amino acid does not migrate in electric field. Why? Because it is electrically neutral, right? The charges cancel out. Whereas at pK1, it is negatively, so it's positively charged, and pK2 it's negatively charged. Chemical environment affects pKa values. Alpha carboxyl groups are much more acidic than normal carboxylic acid groups. For example, glycine carboxylate is much more acidic, at least 100 times more acidic than the carboxylate group of acetic acid. Why? Because pKa of this carboxylate is 2.34, whereas the pKa of the carboxylate from acetic acid is 4.76 or 4.8. Remember, pKa is a log value. So 1 unit means 10 times. 2 units means 100 times. And hence, glycine is 100 times more acidic than acetic acid. Alpha amino groups are slightly less basic than in amines. And in this case, we can compare between glycine and methylamine, right? The amino group in glycine has a pKa of 9.60, whereas the amino group in methylamine is 10.6. What is the reason behind this? To start with, for carboxylic acids, one of the main reason is the presence of a positively charged amine next to it, right? So this is an electron withdrawing group, and it withdraws electrons and makes this carboxylic acid group more acidic, whereas there are no electron withdrawing group attached to this methyl group in acetic acid. What about methylamine and the glycine amino group? The same way as the same explanation can be given for amino group as well, right? It is attached to an alpha carbon, which has a carboxylic acid group, which is an electron withdrawing group as well. And that withdraws electrons and makes this amino group more acidic as compared to methylamine. And hence, it loses a proton at a pKa, at a pH much lesser, or at least 1 unit lesser than methylamine. So the amino group in glycine is 10 times less basic than the amino group in methylamine. Let us consider the titration curve for histidine. This is an amino acid with an ionizable side chain. Now, this titration curve looks ominous, right? It's a little more complex than glycine. But I'll help you understanding as to how you can draw a titration curve if you were just given one structure, right? If you were just given this structure, how would you set up a titration curve? Now, the first thing that you need to understand is that in this molecule, right, that is shown here with +2 charge, has 3 ionizable groups: an amino group, a carboxylate group, and a side chain group, which is an imidazole group, right? So now this group, what it can have, it can protonate. This group can get protonated. This group can get deprotonated. You need to understand that first. What are the functional groups that can get protonated and deprotonated, right? Now, all amino groups are protonated at physiological pH and below that. And all carboxylate groups they hold on to a proton below their pKa, that is probably close to around 2, right? Now, if you remember the table in part 1, the pKa's of all the amino acids, right? Carboxylates have a pKa that is close to 2. Now, this is the alpha carboxylate, right? So any at pH above 2, it loses a proton. So this is the first functional group that loses a proton. Now, if you were to consider a titration curve with pH on the y-axis and OH minus equivalence on the x-axis, right? As we increase the equivalence of OH minus, right? What does OH minus do? OH minus is essentially taking away the proton from each functional group, right? The first group that loses is the carboxylic acid group. As we go from here and we reach this inflection point, right? This is the inflection point. This is the point where pH is equal to pKa for carboxylic acid group, and it happens to be 1.82. Now, as we go, now at this point, this group start losing a proton. As we go up, and again, as we reach another inflection point, right? That point is when this group has completely lost its proton, right? Now, the first species has a charge of +2 because this carboxylic acid is neutral, and 2 positively charged charges on the amine and the side chain imidazole. After pKa1, at pKa1, these two species are in equimolar ratios. At pKa, at pH of 6.0, all of this carboxylic acid has become COO minus. And so a negative charge is added to this molecule now. This negative charge cancels with this positive charge, which leaves with a positive charge on the imidazole functional group, right? And so the charge of the amino acid is +1. Now, at pKa of 6.0, right, you add more OH minus equivalents, and then you reach another point, right, inflection point at pH of about 7.59. That is when this species is in equimolar quantity with this species, where the charge is essentially neutral because the side chain imidazole has lost its proton, starts losing its proton, and it becomes 0. Remember, this is when this group starts losing its proton, right, at pK of 6.0. And at this pH of 7.59, it becomes electrically neutral. Now, at that point, right, you add more OH minus equivalents, and you reach to a point of pH of 9.17. That happens to be the pKa of the amino group, and that is when the amino group loses its proton and starts losing its proton. And pKa of 9.17 is when these two groups are in equimolar quantities, right? At pKa of 9.17, all of the protons of this has been lost completely. Whereas NH2 starts losing its proton, and this gets done at about pH of 12, right? Now, if you look at this carefully, there are at least 3 or 4 inflection points, right? But how would you calculate the PI, isoelectric point, for histidine? And for that, you need to consider the charge first. You need to write down all 4 structures. And how do you get 4 structures? Because you start with the structure where the carboxylic acid has a proton. Always remember that. Start with a structure where the carboxylic acid has a proton. Go to the next structure where the carboxylate is negatively charged. Then the next structure where the group has, whichever group that has a higher pK than the carboxyl group, right? It could be the side chain. And the next structure when your amino group, the alpha amino group, loses its proton. So this is just an example, right? Now the PI is calculated based on the charge. Now remember, PI is equal to pK1 plus pK2 divided by 2, right? So pK1 happens to be pKr because you need to consider this structure for PI. pK1 plus pK2 divided by 2 will give you 7.59. And that is how you calculate the PI. I want you to practice this for other amino acids like lysine, aspartate, glutamate, and cysteine. Please try this at home.